Good afternoon, everybody. Good to see you again. Today's speaker is Brendan. Brendan has been here since 2018, 6 years ago. Brendan already arrived with amazing research experience and quite a few publications. He started to work on Anole lizard. I don't know why, actually, but they're cool. But then he got to go to the Amazonian forest in Ecuador for at least the second time in 2019 and was introduced to a beautiful system with lots of interest from cognitive aspects. And he kept working on this system. And he's still working on it now. He's going to defend in 2 months, and I will let him show you why this is a wonderful system for research. It's all yours.
All right, thank you, Reuven, for the introduction, and thank you, everyone, for coming out to Colloquium today. I'm going to talk about, what I have been up to while I've been kind of shuffling around the department for the past 6 years. So first off, there are a lot of people that helped me accomplish what I have accomplished. This has been an international collaboration between McMaster, myself, and Jim Barnett, who's now a professor at Trinity College Dublin, Justin Yeager, who is a professor at Universidad de las Americas in Quito, Ecuador, my supervisory committee, Reuven, Ian, and Nick, wonderful undergraduate thesis student, Anna Vanit, Lola Garderas, the manager at Centro Hambatu breeding facility that we did some studies with, as well as Todd Swanson, the director of the field station that I work at, and many others. Another quick thank you to the Napo Quechua Nation, who welcomed me into their ancestral territory to perform my fieldwork. So I am a visual ecologist, and visual ecology concerns itself with the way that visual traits and visual systems interact between animals and their environment to influence their evolution. And I'm also a behavioral ecologist, maybe a little more familiar to people in PNB, where we kind of look at how, in a similar vein, behavioral traits evolve between individuals and their environment to influence, reproduction, evolution. So within the field of animal behavior, we often try to break down a lot of the functions into what we call the 4 Fs. So that is fighting, feeding, fleeing, and we're going to stop there because that is where my research is concentrated. And I expand fleeing to kind of just mean enhancing survival. So how do animals use visual and behavioral traits to promote their survivorship in the wild? And in anti-predator visual ecology, we are looking at how visual traits evolve to disrupt the predation sequence. So if the predation sequence is something you're not wholly familiar with, it starts with an encounter between the predator and a potential prey item. The predator must then detect or identify the prey and then pursue the prey. And then it has to capture or subdue prey and then handle prey if there's like a shell or a carapace that needs to be removed. And then consumption finally happens at the very end of that. And so the anti-predator visual phenotypes we'll be looking at today are kind of, we think, evolving to disrupt this kind of early to mid stage of the predation sequence, the detection or pursuit elements. So first off, in terms of anti-predator phenotypes, we are probably the most familiar with crypsis, which is reducing detection. So crypsis is kind of a fancy word for camouflage, and there are a bunch of different mechanisms within camouflage that can help reduce detection by predators. So you can evolve a phenotype that matches the backgrounds that you frequently occupy, like this frog here and this leaf. You can evolve disruptive coloration that isn't a perfect match to your background, but kind of has some color similarity and some kind of sparse coloration that breaks up your outline and helps you blend into your background. And then lastly, we won't be talking about this one today, but I think this might actually be the coolest, is masquerade. So these are satanic leaf-tailed geckos, which have evolved to very closely resemble curling leaves. Another cool example of masquerade is the bird poop frog from Southeast Asia, which literally has just evolved to look like bird poop. So Crypsis brings some advantages, specifically in that it is a very low energy investment anti-predator strategy. You just occupy a place where your Crypsis is functional and you don't have to move for a very long time and you're probably going to be safe. And so this allows you to basically bypass the entire predation sequence. If predators don't detect you, they don't even know if they've encountered you, and so you can bypass the pursuit and everything past that. But Crypsis is not perfect, and it imposes some disadvantages as well, mainly in that it restricts activity spatially or temporally to places where your coloration is suitable. So this toad here would do great in leaf litter, but on this green leaf, it kind of sticks out like a sore thumb. So this is a staged photograph, but this would not be a kind of spatial niche in the environment that this animal would be able to exploit. So it has an opportunity cost imposed upon it by the coloration it has evolved to avoid predation. These opportunity costs can be kind of eased if you use a different anti-predator strategy, which is aposematism, in which you are detected very easily by predators, but instead you are reducing their pursuit of you. And another word for this is warning signals. And it's basically animals evolving bright coloration. And we see this terrestrially, often in terms of caterpillars, and we see this aquatically too, like this lionfish that has quite the vibrant salient signal. So the way that aposematism actually does reduce pursuit relies on the fact that the warning coloration is an honest signal of defense. So a predator, like this blue jay here, will eat a butterfly that is kind of loaded with toxic cardenolides and glycosides in its body, and it kind of realizes something is not quite right, and then it pukes its guts out. And this is a classic example, basically 60 years old. And so animals will either evolve to innately avoid things that look like what it just ate and got sick from, or it will learn through experience. And so either pathway of that we call generalized avoidance, where things that share kind of a very salient cue will be avoided by predators. The way that this works can be visualized by something called the cone of protection, where if we have any trait, like body color, for example, there's a darker end of that continuum and a brighter end of that continuum. And a toxic model species will have a species kind of trait mean in the center, and some individuals will be darker, and some individuals will be brighter. So generalized avoidance means that predators will avoid a range of phenotypes that should encompass most of the natural variation of that warning signal that we see in nature. So predators avoid prey with similar traits. This is then itself exploited by something called Batesian mimicry, which we think of as a form of visual parasitism. So Batesian mimicry is when a non-toxic species adverges onto the warning signal of a toxic species. And this is a classic example here in North America, the milk snake, which is non-toxic, versus the coral snake, which is toxic. And when we talk about Batesian mimicry, we use the word imperfect because it's very rare for a mimic to perfectly quantitatively match its model's traits. And this particular mimicry system is a good example of this because the order of the banding is different between the model and the mimic. And there's an old rhyme that I'm learning is less and less common in Canada for people to know, but it's red touches black, venom lack for the mimic, and red touches yellow, kill a fellow for the model. Side note, that really only works in certain parts of North America. If you take that down to the neotropics, you will get yourself killed if you try to operate by that. And so the reason that we consider this parasitism or an exploit is that the presence of the mimic undermines the model's signal honesty in an environment. So if a predator does encounter a mimic and decides, hey, this thing's bright, but I might sample it anyway, it will think that that individual is safe to eat, and it is. And so that may change how it perceives the model over time. So the mimic kind of undermines the aposematic dynamic that the model has established through generalized avoidance. In terms of how the mimicry itself evolves, there are some competing theories, but the most common one is called the cryptic origin theory, where the mimic species represents a transition from crypsis to aposematic coloration. And so in order for this to happen, the mimic has to be in sympatry with the toxic model species, and there has to be some sort of selectable mutation or process that confers resemblance in the mimic species. So I have a very, very simplified phylogenetic tree here where we have a cryptic lineage on the left with brown-colored frogs that are edible, not toxic, and we have an aposematic lineage with red-colored frogs that are toxic. And so the aposematic frogs have established a cone of protection, or this protected trait space where predators are probably avoiding bright red frogs that look like them. If one of the mimic or one of the cryptic lineage species is in sympatry and the right kind of spark of mutation or selectable event occurs, through many generations is the theory that all of a sudden the one frog from the cryptic lineage is going to look a lot more like frogs from the aposematic lineage. And if it's able to do that and enter the cone of protection and gain anti-predator protection, we then dub this the mimic, the edible species, and the model, the toxic species. So aposematism, real or mimicked, has its own advantages. As I mentioned before, it reduces the opportunity costs of crypsis. So this little blue jeans frog, that is the real name, blue jeans frog, it can occupy basically any spatial niche in the environment that it can physically access because it's going to brightly contrast against essentially everything. And so its anti-predator protection strategy is not compromised depending on where it is in its environment. And what this also opens up the door for is what we call safe channels of communication that can be used in other contexts. So these Atelopus toads evolve a very bright signal with yellow, black, and red that's highly contrasting. So they're very toxic and predators avoid them, but they also use that coloration for mate choice and reproductive purposes and sexual signaling. So you can co-opt the trait that's giving you protection from predators to perform other functions, and that's kind of a very efficient or streamlined way to evolve certain suites of behaviors. So aposematism does have some disadvantages, however. It's a metabolic investment to create the toxins, and models and mimics still face predation threats. So these can come from specialized predators, like this spider here eating a poison dart frog, or through something called taste and reject sampling, which is where predators will kind of be foraging their environment. They'll see something that's very brightly colored, and they'll catch it and kind of snap it up, and they won't immediately consume it. They'll handle it a little bit, see if it tastes bitter, see if they think they should eat it, and sometimes they'll decide to just reject it and let it go, and sometimes they'll decide to consume it. But this is still a costly event for a model or a mimic. And these threats increase the more salient your signal is. So we think that models or aposematic species in general should be balancing pre-detection versus post-detection protection, or pre-detection versus post-detection coloration. So this may force a compromise between crypsis versus predator deterrence in terms of the overall color phenotype that a species expresses. So aposematic phenotypes may still permit crypsis if you can, for example, cover up certain parts of your body that are super bright, or if certain parts of your coloration that are bright at close distance end up just blending into the environment at a far distance. We think that the costs of detectability should be way stronger for a mimic than a model, and that's because they're unlikely to be rejected during one of those sampling events. So this overall leads us to believe that models and mimics should be under a differing balance of selection pressures for detection versus crypsis. And we think that this is a mechanism that may generate imperfect mimicry. So this brings me to kind of the first research question of my dissertation, which is how do mimics and models balance signal salience versus crypsis? And our hypothesis is that mimics should evolve a less detectable version of the model's signal that allows them to remain within the cone of protection while reducing their rate of predator encounters. So the way that this would look like is we have this kind of caterpillar continuum of darker versus brighter for their body color, but this equates to just less detectable versus more detectable. So when we think about what part of this protected trait space that a mimic should potentially evolve to occupy, we think that mimics should be evolving to be on the less detectable end of the spectrum. And to test this, we are using poison frogs as a study system. Other than just being super cool, poison frogs are really suitable to test hypotheses like these because they utilize a diverse array of aposematic signals for protection. So all of those frogs there are actually the same exact species. It's just that those colorations come from different localities. So within one species, you have a ton of diversity, and across species, we have a ton of diversity as well. And their coloration, we already know, is under many competing selective pressures. So status signaling, male frogs will fight for territory, and it's often brighter frogs that tend to do better in those types of kind of agonistic bouts. Mate choice, so intersexual selection, and camouflage. There is a frog here, down at the bottom. And so that's an example of up close, the dart frog coloration being very salient and vibrant, but at a distance, the kind of broken up black and yellow can blend into a leaf litter background and actually give it pretty good camouflage capability as well. And poison dart frogs have also evolved mimicry systems multiple times. So these on the right are 3 separate species: Ranitomea fantastica, Ranitomea variabilis, highland and lowland morph, and Ranitomea sumerzi. And this on the left are, again, the same exact species, Ranitomea imitator, just different sublocalities that occur in sympatry with each of these models. So we know that poison dart frogs are evolving mimicry systems, and so these allow us to test some of these hypotheses. So my mimicry system that I work with is a little bit different. I work with what's called the Aimeriga allobates complex, where we have the model species, Aimeriga bilinguis, that is toxic, and a mimic species, Allobates zaparo, that is non-toxic and therefore a mimic. And just to the naked eye, it does a pretty darn good job of mimicking those spots in that red dorsum. These frogs are both native to the Ecuadorian Amazon, and experienced predators, or predators that have encountered the model before in experiments, will subsequently avoid the mimic. So we know from that that the mimic's mimicry is effective or functional. And so we're going to use this system to test our hypothesis of whether the mimic has evolved a less detectable signal. And to do that, we're going to collect wild specimens of the model mimic, or we already did. We quantified the signal salience in terms of its contrast against the background. So how bright of a phenotype are you, a signal are you, against a natural background that you might occupy. And then we compared detectability to live observers. And so our general predictions are that the mimic's signal components will be less detectable. This anti-predator phenotype has a couple different components. So there's the dorsum, that red patch on their back. There are the spots, the front spots and the rear spots, and there's the ventral coloration. So the model has this bright blue ventral coloration, whereas the mimic's is more of a dark charcoal gray. So we don't really think of the ventral coloration as being well mimicked, but it's still a part of their signal. And so because they have different components of their phenotype that might be visible differently in different postures or from different viewing angles, we also think that the detectability of each species will differ depending on how you're looking at them and whether it's the model or the mimic. So from the front, for example, the model's bright blue ventral coloration is visible, whereas for the mimic, it's this dark charcoal gray. And so that's the kind of dynamic we're interested in there. So in 2019, I traveled to the E-Arena field station, which is in the Napo province of Ecuador, and I captured 20 models, 20 mimics, and 20 individuals of this kind of size-matched cryptic control species. And here's an example of the environment that they're found in, and then here is the frog at its real size, and then here is the frog blown up. So even though they have this bright coloration, again, they are very hard to find at times. So the first thing we did was quantify the salience of their signal, or the contrast it creates against the background. And the way we did that was in ImageJ using color-calibrated photography. So we simulated an animal visual model. For these data you'll see, this will be birds. And you can basically pass each pixel from a region of interest that you draw in ImageJ through your simulated animal visual model and compare it to a background and get a unit called a JND, which stands for just noticeable difference. And it basically tells you how detectable or how differentiable that signal component is against the background. So the short explanation for that is the stronger the contrast, the higher the JND value for that component. And so from our visual modeling, we found that the model's spots create higher contrast. We see that we have our x-axis, which is chromatic contrast, which is contrast from a hue perspective, and achromatic contrast on the y-axis, which is an independent kind of physical property, and that's differences in brightness independent of hue. So things that are further away from the origin in either direction are kind of considered to be more salient. Achromatic contrast might be a little bit more important than chromatic contrast, but we do see that the model is higher in both of them compared to the mimic. And so that does follow our expectation. We also find that the model's ventral coloration has a stronger contrast than the mimic's ventral coloration. But what we found that kind of surprised us was that the mimic's dorsal signal created a stronger contrast against the background. And that's the opposite of what we expected. So these are kind of just physical properties that we found through computer imaging. This doesn't actually tell us anything about observer behavior. So next, we conducted a detection experiment where this is delivered on a computer to human participants, where we had different models of the model, mimic, and cryptic species with different spot manipulations. So we had each of the three species with the model's spots, each of the three species with the mimic spots, and each of the three species with no spots. And so this kind of factorial design allows us to tease out, okay, is the mimic's dorsum more detectable, but the model's spots more detectable? And these icons were presented in randomized order, position, rotation, against randomized natural background photos. And so the results of this detection experiment were that the model's spots are the most detectable, and that is aligned with our prediction. That the mimic's dorsum was more detectable than the model's, which again goes against our initial expectation, but is corroborated by our visual modeling results. And the other thing we found that really surprised us was the mimic's natural phenotype, so the mimic dorsum with the mimic spots, is more detectable than the model's dorsum and the model's spots. So then we wanted to see how viewing angle affects this. So we did another detection experiment with human observers. This is during the pandemic on Sona. And so we had the model and mimic from a rear view, from a front view, a right-facing view, left-facing view, and a crouched view. Again, on natural backgrounds, but instead of these kind of aerial background shots, these were kind of profile shots at ground level up against piles of roots or a fallen log or any kind of terrestrial surface where we would expect to see these frogs. And what we find is that we have an interaction between angle and species, indicating that the detectability does depend on the angle for the model and mimic. Specifically, the mimic is more detectable from the rear, and the model is more detectable from the front. So this is aligned with our visual modeling data, where the model being more detectable from the front we think is probably due to that ventral coloration being much brighter, and the mimic being more detectable from the rear is probably due to that brighter dorsal coloration. So takeaways from this first segment are that the model's spots and ventral coloration are more salient than the mimics. but the mimic's dorsum and its natural phenotype are more salient, and that was kind of opposite to our expectation. And we have this differential effect of viewing angle and posture for model versus mimic. Overall, this, I would say, is pretty weak support for our cryptic mimic hypothesis, namely because the natural phenotype of the mimic was more detectable than the natural phenotype of the model. So that one kind of has to go out the window. So with this, we find that imperfect mimicry can produce mimics that are more detectable than their models. And this suggests that there's a different balance of selection pressures between models and mimics, and potentially between the different subcomponents of their anti-predator visual phenotype. This could also be due to physiological constraints. So if the mimic simply can't evolve spots that are as bright as the models, it could need to evolve a brighter dorsum to reach some sort of perceptual threshold to trigger generalized avoidance in predators. We don't have a way of testing that right now. It's just a speculative theory. The dorsum may also be sexually selected, and that may have helped kind of accelerate the development of that cue. Sexual selection in general is thought to be implicated in the evolution of aposematism and mimicry. And I'm very interested in specifically how this cue develops, because when we were doing this first component, we were only using adults. And the reason for that was because the mimic coloration as juveniles is non-mimetic, so it doesn't match the model. So this is a juvenile mimic, and it has this black back with kind of a tan ring around the front of its face going down its body. And they only reach the mimetic phenotype when they're adults. So this is something called ontogenetic color change, which is a change in coloration across lifetime. And we think that this may be a transition in anti-predator color strategy. And there's some precedence for this in something called a panic moth. So their larvae in the first instars of their life are cryptic, and then they have intermediate instars where they're starting to turn a little bit orange. And then in their last instar, before they metamorphose, they are conspicuous. They have toxic regurgitative kind of goo that they'll throw up during predator encounters. So we know that some honest signalers can change their coloration from cryptic to aposematic. But we're kind of seeing this in a mimic species, which is unusual. So as a baby metamorph, they kind of look like that, again with that tan yellow ring. And then as juveniles, that yellow ring starts to turn kind of a coppery orange red. And then that red coloration begins to fill in the dorsum as they're adolescents and subadults. And then finally, they reach their adult coloration with the full red dorsum. The yellow spots are developed. So it's improving its resemblance to the model over time. And again, here's just another shot of a model. As a baby, this would just not be confused for this under any circumstances. But as an adult, again, their fidelity has improved quite a bit. So what this got me thinking about was, does this change in coloration signify a change in anti-predator strategy, or is it forcing this compromise where better model resemblance is met with worse crypsis? So is the young stage mainly working on camouflage for anti-predator protection, and then the older phases working for this aposematism that we already know due to the previous research is functional? So my approach for this is I collected wild mimic juveniles of different sizes. They start out super tiny, like 6-7 millimeters long, and then quantify the mimetic fidelity across development, and then compare the detectability of different color stages against natural backgrounds. So to do this, I went back to the Amazon. This is my field site. And I caught and photographed 85 mimics of different developmental stages. And this is kind of what we get for that. Again, we have juveniles. This one's already got a little bit of orange going on in its ring. And then the orange slowly spreads down the body until the full adult phenotype is reached. And the way that we kind of quantify this developmental color change is, instead of computing the contrast between the dorsum and the background, we are computing the contrast between the dorsum or the spots of a mimic individual against a sample of model individuals. So now our visual model is telling us not about how brightly something contrasts against the background, but how something differs in brightness from the coloration region of the frogs you're comparing it to. So basically, a lower contrast for both color and luminance means higher fidelity between the mimic and the model. So we computed contrasts for the dorsum by taking 3 evenly spaced, evenly sized regions of the dorsum and kind of combining them into one dorsal signal, and then computing a contrast between the dorsum of the mimic versus the dorsum of the model. And we did that as well for the front spots, averaging the right and left together, and averaging the left and right rear spots together as well. And so on the x-axis, we're going to have body size, and the top row of figures will be chromatic contrast or color difference between mimic and model, and the bottom is going to be luminance contrast or brightness difference between mimic and model. So we find for the dorsum that the mimic starts out with kind of poor fidelity, a high contrast, and then it has this kind of asymptotic, maybe quadratic function where as it grows, it improves its color resemblance to the model. We don't really see a trend in terms of brightness, so it seems like the difference in brightness that we found in the first experiment between mimic and model is pretty fixed across its development. The front spot data, honestly, was kind of a mess. We didn't find any sort of ontogenic trend, but what this tells us is that the difference in coloration and brightness in the front spots of the mimic is pretty constant across its lifetime. And for the rear spot coloration, we find this similar kind of semi-quadratic nonlinear decrease in contrast or improvement in fidelity with body size for the color contrast, but the luminance contrast is kind of a mess again. But from this, we would say the mimic improves its dorsal color matching of the model over time and its rear spot color matching of the model over time. But this is only cross-sectional sampling. I caught each model once at one point in its life and measured it once. So we've had reviewers tell us that you could have just found a spurious size-based trend, and this isn't actually like developmental or ontogenetic trend or change. So what we decided to do was replicate this with a longitudinal captive breeding study. So I transported the frogs from my field station down in the Amazon, about 4 hours to Quito, to the Centro Hambato de Conservación y Investigación de Anfibios, which is a conservation and research and educational outreach facility. And so there we established breeding groups where we produced F1 clutches of mimics and models. Unfortunately, the breeding tanks had multiple mating pairs, so we weren't able to track kind of parental effects. So we won't have any kind of parentage reveals in our Maury moment. But what we instead did was we photographed the clutches. We kept the clutches separate and photographed them every 2 to 4 weeks, and then did a within cohort analysis of ontogenetic change in these color contrasts. And so we compared these guys to a selection of captive bred adult models. And so what we found is, again, we have the chromatic or color contrast on the top and brightness contrast on the bottom. We found that the mimic improves its dorsal color matching. So this is a generalized additive model. So it's a little grabby onto data noise, but this generally shows us that there's a decrease in contrast or an increase in resemblance over development. And that kind of aligns with what we had found from the field data. From the captive data, we actually see an improvement in dorsal luminance, which doesn't match what we found from the field data. And the model, we were able to test for ontogenetic color change in the model as well. And we see a much cleaner curve for both color and luminance. So the model improves its dorsal color and dorsal luminance resemblance to itself, meaning there's a little bit of color change that happens in the juveniles, but it occurs very quickly. And so they're kind of achieving their adult aposematic phenotype pretty early on in their development. We see no improvement or change in color or luminance contrast for the front spots, which does align with our field collected data. And again, we find this very steep curve in resemblance for the model in both color and luminance for the front spots. For the rear spots, remember this is one that we had found evidence of improvement over time from the field data. So we find that the rear spot luminance improves. That's not what we found from the field data. And the mimic actually weakens its resemblance because the contrast is increasing in terms of the rear spot color. So this is kind of a bit of a puzzling result. And then once again, the effect is less pronounced, but thematically it's still there that the model quickly improves its rear spot color and luminance resemblance over its development. So this tells us that the model juveniles kind of rapidly achieve their aposematic phenotype through the earliest portion of their development. And the mimic gradually improves its resemblance in dorsal and maybe rear spot color. And so what I'm interested in is how does this change affect their detectability, again, to observers? And is this signaling a potential trade-off between crypsis and aposematism? So we're going to focus on dorsal coloration because spatially it's the largest signal component, and it can't be concealed through any kind of defensive huddling postures that they do in the wild. So we did another detection experiment where we looked at juveniles with the yellow ring phenotype and a red ring phenotype. And here's an example of individuals that kind of fit these models. And mature phenotypes with the red ring and dorsum that's kind of half filled in, like for a subadult, and then the full red dorsum for a full adult. And again, we presented this on natural leaf litter backgrounds, and some people in the crowd actually took this experiment, so if you did, thank you. There is a frog on this background. It is right here. And our prediction was that mature phenotypes, i.e. the subadult and adult, will be more detectable than the juvenile phenotypes, i.e. the juvenile yellow ring and the juvenile red ring. And so what we find is kind of aligned with what we thought, but also very much not. So they begin this yellow ring phenotype, which again is the kind of brand new fresh metamorph phenotype, was actually the most detectable of all the phenotypes. But then once it has transitioned to the red ring, that greatly decreases its detectability. So according to these data, the mimic is metamorphing into something that's very detectable, but then its detectability decreases. And then as that red ring fills in to become more kind of just spatial coverage of the red color, then its detectability increases again. And when you're a subadult or an adult, your detectability isn't that much different. So what might be driving the early phase detectability is instead of contrast against the background, internal contrast within its own body. So just the border between the black dorsum and the yellow ring creates a contrast that's very strong as well. And just the takeaway that the red dorsum does increase detectability. So from this, we find we can quantify the precision of a mimic's resemblance by using the same visual modeling techniques instead of to model contrast against a background in your natural habitat, contrast against a thing that you supposedly have evolved to resemble. We find that the mimic improves its resemblance over time, and that our dorsal color change replicates across field and captive cohorts. We also see through these data that the model seems to establish its signal earlier than the mimic does, and that the mimic detectability changes across lifetime in kind of this U-shaped trend where its crypsis is compromised by its aposematic resemblance. We think that size has to play a role here. So when they metamorph into that highly detectable black and yellow phenotype, they are the smallest that they'll ever be. And in our detection experiment, we kept the frogs fixed at adult size. So it's very possible that that plays a role in that color change, and that that same yellow ring phenotype, which we also see in just straight up poison frogs, would not be tenable for a non-poisonous frog to have. So that was all very visual ecology oriented. Now to bring it kind of back to the behavior sphere, I want to look at how behavioral transitions might be timed with mimetic resemblance. So the mimic is gradually approaching this defended phenotype, and it becomes more detectable. So does this mean that behavioral phenotypes change in development as well to kind of compensate or complement its change in anti-predator protection status. So we're going to look at boldness, which is the propensity to engage in potentially risky behavior, and activity, which is a little more self-explanatory, just how much do you move around in your environment. So this is field research that I did in the summer of 2023. I caught 91 mimics in various life stages and photographed them and tested their behavioral phenotypes. And to test their behavior, I used something called a novel environment test, which is this illuminated, camera-equipped arena adapted from Melissa Pena, who's a postdoc in the Ringler lab, who studies a very closely related Alabates species to our mimic species. So this is kind of verified to produce good behavioral data in this system. And so we performed these 15-minute long video recorded trials once in the morning and once in the afternoon for 2 days. So frogs had 4 trials total. And so the way it works is the frog goes into an acclimation chamber where it sits for 5 minutes, and then a trap door that's from a string outside this opaque walled arena is pulled up, and the frog is presented with this brightly lit arena, and we video record its behavior and kind of count the number of squares it crosses, look at how much ground it covers. So our boldness measures are the binary variable of did it enter or not during 15 minutes. And if it did enter, the latency at which it did. And for activity, we counted the number of floor squares crossed in the arena. So there were 5 centimeter by 5 centimeter square grid on the floor. And so by counting the number of squares it crossed, you get a rough measure of how active or how much it moved around. So one other thing we did for these behavior trials was to manipulate the light level. The light intensity that these frogs are subjected to in their visual environment fluctuates naturally, both spatially, like as a function of this canopy gap here, where we have this super bright, harsh, direct sunlight on some parts of the understory, and right next to it, just like perfect flat shade, or just temporally with weather shifts or just changes in the daylight environment. And so this should have consequences for prey species in that there should be increased risk in brighter areas. And we think this should be especially costly for mimics that are utilizing a bright signal but are dishonestly signaling. So we put, of those 4 behavioral trials that the frogs went to, they were randomized into 2 low light and 2 high light trials. So their first trial type was randomized, and then they were counterbalanced from there. So our predictions for this segment were that larger mimic frogs will have a higher dorsal color resemblance, or in other words, that we will once again replicate this dorsal color curve that we found in the last field season and in the captive breeding study. Second, mimics with higher dorsal resemblance should be more active in the environment, and mimics should be less active in high light than in low light, and that mimics with higher dorsal resemblance should be bolder. So they should be entering the arena in a higher proportion of trials, and they should be doing so more quickly. So our first result was that we did indeed replicate our color change. So this is a blown up figure of the dorsal color contrast as a function of body size from 2022. And we found a very similar looking curve for 2023. So that's always reassuring when you find the exact same thing 2 years in a row with field data. What we found was that there is no effect of dorsal color resemblance on activity, which doesn't align with our prediction. But we find that the frogs were less active in the high light treatment than they were in the low light treatment. And so that does align with our prediction. We found no effect of dorsal color on boldness, either in terms of entry probability for the arena, or for the ones that did enter, the latency at which they did so. So that again goes against what we were expecting. But we did find that larger mimics are less bold, both from an entry probability perspective, where frogs of a larger snout vent length, which is just a general body size measurement, were way less likely to enter the arena compared to smaller frogs. And the latency at which they did enter was longer for large frogs that did enter the arena. So they're less bold, both in terms of their probability of entering being lower, and the latency at which they do enter is higher. We also found that frogs are less bold in the afternoon, which is another term that we had in our model. So in terms of replicating our dorsal color curve, we are now very confident in saying that the juvenile and subadults undergo this kind of substantial phenotypic shift that we are considering a transition from crypsis to aposematism. And this puts a new twist on the cryptic origin theory, because instead of this change happening where the mimic lineage starts cryptic and ends aposematic, instead of happening across many, many generations, it's happening within the lifetimes of single individuals. So instead of having to endure tens or hundreds of generations in the middle where you're no longer good at crypsis and you're not yet good at mimicking, instead of having a bunch of maladaptive kind of time there, you may only have to endure a couple months of maladaptive time for each individual. So just another way of thinking about how mimicry evolves. And this is a testing ground for proximate mechanisms of like literally how does the color develop in these frogs. Is it dietary? Is it genetic? It's probably some combination of the two. We also found the lighting environment influencing the activity in our frogs. Specifically, this was independent of time of day. So whether it was a low light treatment in the morning or the afternoon, time of day didn't affect it. It was all with the lighting environment. So we think this increases their exposure to predation, where a brighter environment highlights their imperfect mimetic phenotype. There's other data from anolis lizards, actually, where brighter environments make distinguishing between reds easier, and that would be very relevant to this species. And this reduced activity has implications in other contexts. So human activity in these frogs' habitat is causing a lot of deforestation, so there's more canopy disruption, more and more happening in their environment. And this has consequences for things like their reproduction, where this is a male mimic, and the males travel long distances, hundreds of meters sometimes, to transport tadpoles to suitable bodies of water for them to develop in. So if changes in the environment affect their activity patterns, this could then affect their reproductive patterns as well and have kind of spatiotemporal consequences in their evolution. As far as what we found for body size versus boldness, where larger frogs were less bold despite having kind of a better warning signal, it's possible that this is just a shift in the suites of behavior that the species is using, where the juveniles might be in a growth stage where it pays to be very bold, entering lots of different environments, trying to find food and grow. And then as you're an adult, you kind of slow all of that down. You're ready to reproduce. You don't need to take any risks. Just find a territory, hold it, call for some females, and hope that you get lucky. A couple other possibilities are that it's a selection gradient where we were catching lots of bold mimics, but those same mimics would be kind of predated upon or just die in their environment and not make it to adulthood. So I could have just been capturing a selection gradient there. And it could also be that mimics simply cannot afford to increase their boldness like we thought that they would, even with their increasing salience of their warning signal. Just because it's a dishonest signal, you can't actually afford to modify your behavior to try to exploit that any more than you already are. So the overall take-homes from what we've seen today are that aposematic signalers do incorporate crypsis into their signals, and defensive coloration strategies are temporally flexible. So the strategy you use as a juvenile may be very different from the strategy that you use as an adult. We have found that there's a very complex relationship between aposematic phenotypes and mimetic resemblance, specifically with us finding a little bit of a mismatch between our field data and our captive data, which again could be due to things like diet. It's expanding our understanding of cryptic origin theory for the evolution of mimicry, where again we have this transition happening not in hundreds or thousands of years of evolutionary time, but within a few months of the individual's lifetime. Environmental fluctuations influencing animal behavior, that's not horribly new, but this species has some pretty cool ways that that can impact their ecology. And just a principle that I think is good, given some of the events that I experienced during my grad school, you should seek to design your experiments in a way that they can at least partially kind of corroborate or replicate themselves. I think that's pretty important to do. Future directions that I think someone else could take this in is examining predator decision-making and mimic fidelity through machine learning and image processing, because at its core, Batesian mimicry is exploiting a classification error, where a predator or an observer is processing a signal and misattributing it to something that could cause it harm as opposed to something that it can eat. And so you could use something like a support vector machine. So these frogs in this area of trait space would be unsafe, and these frogs in this area of trait space would be safe to eat. I think you could do some interesting things there. I didn't look at ontogenetic behavioral changes in the model species, specifically because for this model species at least, I couldn't find enough juveniles to have a good sample size to do behavioral trials with. For some reason, I could find as many mimics as I wanted, but the models I had way tougher time with. So if someone else had a better model species that was very abundant, or they just had access to a larger sample size, I think an individual that's changing its strategy but being an honest signaler where it does have a chemical defense, I think there you could potentially start to see some behavior shifts. So what you can do is you can manipulate the defense status of frogs by provisioning their food either as tadpoles or as early juveniles with chemical defenses in captivity. So I almost made it through a whole talk about frogs without mentioning that I have pet poison dart frogs, but I do. This is a photo of one of them. This is a Ranitomea variabilis, and their reproductive ecology is very cool because the moms will sometimes lay eggs for their tadpoles to eat. And those eggs in other species have been found to carry the toxins that the frogs later develop. So those tadpoles are kind of getting pre-laced or pre-loaded with defense compounds. And at least in other species, they then metamorphose with a low but functional level of that chemical defense already established. So if you can manipulate the diet of tadpoles of a species like this that naturally kind of some moms feed eggs, some moms don't, it's a facultative egg feeding, I think you could start to do some pretty interesting experiments with that. Last thing before I let you go, we're going to take a quick quiz. So A, are both of these frogs toxic? Or B, are both of these frogs non-toxic? Or D, is the left, non-toxic and the right is toxic? Or C, is the left toxic and the right non-toxic? Andre. Which one? This left non-toxic? You are correct. So very nice job. Yeah, this is a mimic. This is a model. The way that I tell in the wild is this border here, this kind of dorsolateral ring line, is way stronger in the mimic, and it serves as a pretty neat barrier between the red dorsum and the black torso, whereas this picture doesn't do a great job, but the models kind of, it transitions from the red to the black in a more diffuse way. And the spots of the model are also just ridiculously brighter than the mimics. They're super bright. Once again, I have a lot of people to thank. So I would like to re-thank the people that are on this slide, as well as thank just the department and university. Despite some of the stuff that happened during my grad school experience, I actually had like an amazing time being at McMaster and in this department. And a lot of the people in this room are a big reason for that. So yeah, very appreciative for all that. Thank you, thank them, and yeah, happy to take any questions. David.
In your visual search experiments, you're using a human eye that has 3 cones, and their normal predators are birds that have 4 cones.
Yes.
So did you think of altering the image so it looked to the human like it would look to a bird?
So that's a very good question. And yeah, it's important to kind of taxonomically contextualize what's going on. So we've done analyses. I didn't show the data. If you analyze the spots especially, the spots do have UV information in them. But if you analyze the contrast against the background with the spots with and without the UV, there's actually no statistical difference. So whether an observer that is looking at it with ultraviolet sensitivity or not doesn't seem, at least from a visual modeling perspective, to change the detectability. Sigal.
Really you. What a predator sees depends on where the frog is.
Yeah.
So if they have a propensity to kind of hide under the leaves or twigs or whatever, they're not going to see that area that you thought was so obvious. Where do these frogs hang out, and where do they get taken? From the back, from the front?
That's a good question. So the frogs, they are leaf litter dwelling. So they are kind of just out and about on top of leaves. They'll go under leaves. Males, when they're calling for a female, they will go up on like a fallen log, something that gives them a little elevation for their call maybe to travel a little bit better. But so I do see, I think one of the frogs in this presentation actually had it. I find mimics with like deep gouges and scars in their dorsum that I'm assuming are from like near-miss predation events. So I think that they're like, they can hide, but they're clearly having predation encounters as well.
And do people know that males are definitely taken at higher rates than females because of their signaling behavior?
No, I don't know whether that would be true. I haven't seen any data on it. I haven't collected any. What I will say is it's actually really hard to catch a male while he's calling because he's very vigilant. So they'll get up on that log and I'll try to catch it and it'll just dart under the log. It usually will be up on some sort of like structural refuge. And then I'll have to wait like 10, 20, 30 minutes for it to come back out and start calling again. And then I have another chance to get it. But when they're in kind of calling mode, it's pretty hard to get them. I have much better luck when I'm just out in like a flat, even part of leaf litter, and you kind of like walk around and shuffle your feet a little bit, and then it's very rare to just see one motionless. It's when they start moving, that's when you see them, and then it's kind of like a chase. So I would say if I'm a predator, I catch them more in like open foraging area compared to on calling perches. Jesse.
Yeah, so I'm curious more about like how you catch them and to what extent sampling bias is a concern. Because factors like the frog's boldness and its activity level and its contrast makes you more or less likely to catch certain frogs than others.
Yeah, that's a very good question. So I would guess that at the whole experiment level, I probably, if there's a bias, it would be that I'm catching brighter individuals and I'm catching bolder or more active individuals. So if that were the case, then I would say the estimates, like the true global value of the estimates, you could probably bump them down a teeny bit. But I don't have, because for the behavior studies, I only tested the mimics, so we wouldn't get into like a mimic versus model differential selection bias thing. And I don't really have a reason to believe that it would affect the two species differently for the visual modeling data. I probably just may have overestimated some of the brightness if there is that bias.